trying to salvage as much of this stuff as they can and take it away, but they're actually going to come back after we've left the area and plan to take it again. No. Yeah. Well, let's keep away from that. So a slight complication to our plans. We decided we would come and actually visit the vent sites, look for diffuse flow near those. We almost drove into a tripwire from some of the cable that's been left behind from the Shinkai visit to the same sites earlier in the month. We know from Kentakai that they've actually been back to a couple of times for dives where they've been trying to clear some of this material away and drag it off to different parts of the vents, but unfortunately there's still some of it present. It may just be caught in that crevice and has snapped, yeah. So if we go around this lump to the right, maybe we'll get well away from it. That was, yeah, that was 4959 was the top of the cable. 4959. Okay, we have some nice targets ahead of us. We just want to get in close enough and see what's there, but if there are shrimp living there, that may be the kind of temperatures we want. So let's just... There's some nice flow in that crack right there. Should we try that?
Yeah.
Apparently they didn't get it all. Ken wrote to me last night. He said they actually dropped about 5,000 meters altogether. There's actually a few targets, Casey. There's a little crack just here that's bare grey rock without the shrimp in it. This looks more vigorous than we saw before, doesn't it? Yeah. Actually, this isn't the Shrimp Gully site. That's a different mound. No.
Oh, which one is this?
This is the one. This Max named this the other day. This is the one. That's a different mound. But wow, it's like a blizzard here, huh? Mm-hmm. you can find anywhere in this area where you can set up, none of it's going to be upsetting to Julie. Okay, welcome back to the RV Falcor. The time is 1:30 in the afternoon, local time, which is the same as East Coast in the US. We've been on the seafloor just a little shy of 4 hours. Our primary objective for the rest of our dive has been to collect some really good filtered water and fluid samples for Julie Huber for our microbial studies. We wanted to get temperatures in the tens of degrees up towards 100 degrees Celsius. And we found a pretty good location here, but it's teeming with fluid flow, teeming with shrimp, but there's sufficiently large amounts of the fluid flow that it's actually blowing the vehicle around. We're just trying to take the time to work out how to get the vehicle set up. We are at a depth of 4,959 meters. We're on top of the same mound that had the high temperature black smoker chimneys we were sampling a few days ago. I think 4 or 5 days ago we were here. And we'll just stand by as Casey sets up as pilot to get things settled out and ready to do the sampling. What a fantastic sight. This whole hillside is like a valley alive with fluid and shrimp. I really hope this is better than 10 degrees Celsius.
It is. No matter what, I'll take it, so.
It's that time of day.
It looks just like that black opening in the back looks just like shrimp.
Yeah.
12, 14, 16, 17, 18, 19, 20.
Shall we zoom?
Can you? Yeah, that's a good idea.
See that black, also a crack up into the right.
23,
Probably get some of each.
24, 23.9, 24.7, 25, 26.2, 26.5, 25.5, 24.3, 22, 18, 17, 16, 19, 20,
Let's come back closer. It was 25 down here. This is almost like a white milkiness.
21, 19, 19,
Right, just down the bottom there to the left.
17,
See that dark crack just there?
Wow. It's just a wash.
What do you got there, Sean?
11,
Huh.
It looks like a mango.
11.8, 12, 13 and a half,
Push it in.
14 and a half, 13, 12 and a half, 11, 12, 11, 12,
Can you zoom back out for a second?
13.6, 14,
If that's okay.
15,
Yeah. Can we switch cameras back out, Casey? Get a bigger field of view.
18,
8 up there.
Oh, well done.
23, 24.5,
Should we pan up and zoom back in on that one? That looked promising.
23, 22, 17.
So we switch cameras back, Casey? Yeah, it looks like there's a little rill just about there of flow. Might be under a little underhang or something, overhang. Go left from there.
11 and a half, 10,
It's hard to be tall.
14, 15.7, 17, 18,
Go left.
20 and a half,
And it's going to be probably about
21,
15.
22,
Let's go a little left.
22.3, 22.5, 22.4, 22.5, 22.4, 29, 23, 23.3, 24, 24.7, 25,
Keep going left.
26, 27, 28 and a half, 29, 29.4, 5, 30,
Just keep going.
8 feet climbing, 31, 31.3, 31.4, 31.6, 27,
Can the camera go left, Mario?
7,
Or hold it.
32, 33.5,
get that while you're happy.
I mean, it's really steady
Oh, cool.
over days.
Yep.
Yeah, it's very steady at about 32 and a half.
Okay, let's
Just dropping a little
set up for that.
Did you lock it?
Yep.
Alright. Start with one. First one, right?
Ready?
Yep, we're gonna start purging.
Sample
What's this? We're just. It will
just
be
purging.
before, but we'll purge it. It'll have it. Hurry up now, here, 1:30.
Yep.
We leave about 2:15-ish, 'cause it's 3 hours, 45 minutes last time. We'll check the number real quick, so half an hour.
Do we have a sample number for this one?
Yeah, it's sample 19.
Sorry.
So we can get one complete suite of samples from here.
Yep.
I'm afraid we're out of time. Sorry. It's I really thought the that hillside was going to have a lot more warm temperature samples.
It's okay. This was a good sample.
And what are we on?
Solid
57% battery.
Yesterday we sampled
That
like
hurts.
a solid 29 and it was really sticky.
Oh, good.
Yeah.
Stinky being the biologist word for it.
Delicious.
Stinky delicious. Ay, caliente.
Yeah.
So the fluids we're sampling here from this is it's like a rocky valley just festooned with fluids and shrimp, and the fluids we're measuring here are about 32 degrees Celsius, which is actually about the same kind of temperature, I think, as the surface ocean here in the tropical Caribbean. So these should probably be quite happy. Well, let's hope they're going to be happy. They should be happy. I'd like to live here. It's pretty.
Okay, we're gonna start pouring for.
Starting Staravex number 4.
Steravex number 4.
Gone in like a dive.
Great. Is Julie still here? She's going to...
Just woke up so excited she ran away.
Well, you know, with all these shrimp here, we should probably go get Cindy and let her come and tell people about what they're looking at. So I'm just going to go find Cindy in the library, and we'll be right back.
Ladies and gentlemen, may I introduce in a moment for the first time live on RV Falca telepresence the one, the only Casey Machado. Live from the Nereus pilot seat.
Good afternoon, everyone. This is probably the first time you've heard my voice, even though you've probably seen many of my actions going through already. Right now we're just set up, taking some of the super samplers for Julie. This site is a little bit difficult to pilot through because there's so much relief in the terrain. You're constantly going up and down. There's a lot of boulders everywhere. So for those of you who aren't familiar with what Nereus looks like behind where all the cameras are, she sticks out pretty far, and there's lots of overhanging parts of Nereus. And then the bottom of the tool sled is actually really flat. So it's particularly difficult to set up on a site if you're set up and you just land that flat bottom on top of one rock, you end up being a foot or two higher than you want to be. And then if you could see all the other cameras that we have here, they're sort of looking out to the side, and one's going forward, and I have views out to both sides. And right now we're pretty much in a little bit of a hole, and there's walls sort of coming up at about 45-degree angles, sort of on all sides of us. So it makes for a little bit of a difficult setup, but nothing that's beyond my capability as a pilot. So I tend to enjoy being challenged with these sorts of things.
So Casey, what's the challenge of Nereus compared to Jason then?
Real temperature.
So Leighton just asked a very good question as what the differences are between piloting a more standard ROV like Jason versus piloting the more specialized hybrid ROV breed of vehicle such as Nereus. And say that vehicles like Jason are much more workhorses of the ocean robotics community. Jason has probably I don't know, about 4 times more thrust than Nereus in all directions. So when you want to go somewhere, you have the authority to do so pretty readily. And the place where you notice that the most is typically in the vertical thrust direction. So if you have a lot of vertical force that the vehicle is able to generate, you don't really have to pay as close attention to ballasting of the vehicle. Whereas in operating Nereus, we probably only have about 100 pounds of vertical thrust, so we have to make sure that our ballasting is spot on so that we aren't using all of that thrust to hold us up or all of that thrust to hold us down. We try to shoot for being a little bit heavier than neutral, and if I'm a little bit heavy on the bottom, that lets me sort of drive up to a site and then just let the slight weight of Nereus, it's usually about 5 pounds heavy, let that just set down in the site in front of me, and then that way you don't stir up a whole bunch of dust, as you've seen me do numerous times, I'm sure. So whenever you see that giant dust cloud, that means I'm doing a lot of maneuvering, a lot of thrusting up, and it just kicks up all the sediment behind me. And then depending on which way the currents go, sometimes it comes in front of us, sometimes it doesn't, we get lucky.
All right. Okay, this is Cindy Vandover from Duke University. I am invertebrate zoologist, ecologist, specialist in studying deep sea chemosynthetic ecosystems. And for those of you who are listening in, I'm about to tell you a little bit about the shrimp that are here. If you've been paying attention the past few days, you've already heard much of this story, but there's some fast-breaking news coming from the dissection laboratory, which is right next to the dustbins on the ship. But we have been collecting shrimp and taking pieces of tissue for genetic studies. But we've also been working with Max Coleman and thinking about trophic interactions in these sites. We have 3 species of shrimp here, we think. And we're under the impression that the shrimp you see in the video right now were relying on chemosynthetic bacteria directly, grazing on chemosynthetic bacteria for their nutrition. What we found is, at least from the shrimp from Von Dam, the shallow site, when we open up the shrimp guts, so we were going in dissecting for other tissues, and decided to pull out some stomachs and look at what the stomach contents are in these shrimp. And I did 5 shrimp last night, opened up the stomachs, and they are feeding on other shrimp. And one had a snail inside its gut. So it's actually remarkably easy to identify the body parts of snails and snails and shrimp in the guts of these swarming shrimp. So that changes the way I think about these animals. So the gut contents we looked at were from the sparse shrimp that Max has been interested in. And so I think today it looks like we might have a chance to pick up some of the shrimp here. And I'll do some more gut content analyses to go along with the stable isotope work that Max is doing, and really try to sort out what the average shrimp is eating here at any given moment. The dissections we did last night, we're parsing these little shrimp. They're very valuable. It takes a lot of work and effort to get them, and so when we get them on board, we all start to think about how to use them best. So last night I think I pulled off the most pieces of shrimp that I ever have in my life and distributed it to different people. So for Max Coleman at the Jet Propulsion Lab, we cut off the brachiostegites, which are the inflated bits of carapace that, if you look at this live video, it's the white patches on the sides of the shrimp. So I just slit that carapace, and Max took the tissue on the inner surface of that carapace, which typically is covered with bacteria, and what we thought the shrimp were relying on for the nutrition, along with some of the bacteria on their mouth parts. So that was for Max. And then for Julie Revio, who you heard from earlier today, a postdoc in Julie Huber's lab, I picked off pieces of the mouth parts, in particular a piece called, lovely name, called the scaphonathite. So she now has some scaphonathite tissue in vials that she's preserved in glycerol, I believe, for some genomic work. We also, for Julie, collected hindguts of these shrimp. So basically the intestine, or what, if you're eating a cocktail shrimp, what we call the vein of a shrimp. It's really the gut of a shrimp, hindgut of a shrimp. So she's got that also with the idea of sharing with collaborators that tissue to look for symbiotic bacteria resident in the guts of the shrimp. Let's see, then I went on to collect hepatopancreas. So the hepatopancreas, or liver of these organisms, is diverticular outpocketings of the digestive system, where main part of digestion takes place. Collected pieces of that, froze some of it, and put some in 70% ethanol for Chip Breyer's lab. Chip is the scientist at the Woods Hole Oceanographic Institution who's interested in the metal composition of the tissues. And what we want to do is compare these Piccard shrimp, which are smaller than the Von Dam shrimp, and we're interested in why the shrimp are stunting. Oh, look at that guy go. I think I think we might see Wild Kingdom. Wow, go get him. Go. Oh, I think.
That's the one you said you wanted.
That's the one I want. That thanks.
So, one of the ideas that's emerging is that maybe these sparse big shrimp are a different species from the small shrimp, and the small shrimp might be the bacteriivores, and the bigger shrimp there is a carnivore. So look at him go. He's on the hunt. Get him. Get him. Get him. All right, guess he's taking a pause. Anyway, so hepatopancreas, that lies underneath the carapace, right, part of just off the stomach, feeds into the stomach. And we'll look for metals in that. And then, let's see, also for Chip, I wanted a control tissue that I didn't think would have so much metal. So we gave him a couple of pieces of abdominal tissue. And then I took my own abdominal tissue. And then also the gut contents. So by the time we were through, almost every little piece of the shrimp was set aside for research back in our home laboratories.
There's a shrimp above that one that looks like it's attacking.
We're staying with this one. This is our horse. I mean, there's definitely the big one.
Yeah. So the other really fun thing about these shrimp, if you haven't heard already, is that they have placed their eyes on their backs. So instead of eye stalks and black beady little eyes, they have, I think he's just bumbling around. I don't know. You know, an alternative is he could be amorous.
Oh, he wants to.
Because they have the male passes to the female as spermatophore. But
So he's either angry or
Is that-
horny.
yeah. What's not to love about a shrimp?
You can't really see your intake. I mean, I think it's right there.
I think it's coming back up.
So these shrimp, swarming shrimp, have been given the name Remicaris hibisi. Hibis was the original, not the original, was the ROV, the remotely operated vehicle that discovered this site and took the first pictures of the shrimp. And I think even collected some of them. I'm sure collected some of them.
Go down. That one's.
Now it's possible, we know there's a shrimp called, well, we know there's three shrimp here. There's Lebius, which is really easy to tell apart from the others because it's got a green gonads. And then there's Alvinocaris, which is a big bruiser carnivore. Maybe that's what we're looking at here. But Alvinocaris has a long rostrum, and I just can't make out with this video whether this big shrimp has a long rostrum. That could be possible. And then the littler shrimp are Remicaris hibisi.
Yeah. 32.5.
Stopping the filter at 1805.
The anemone you see on the right of this
Switching over to
screen.
bottle number 5.
Oh, thank you, Casey. Those anemones, we believe, are shrimp-eating anemones. So, and the reason I think this is they resemble the anemones we find in deep water on the Mid-Atlantic Ridge. Oh, that's beautiful. On the Mid-Atlantic Ridge at the TAG hydrothermal site. And at that site, I was in the submersible Alvin, and the pilot saw an anemone, the tentacles reached out, or a shrimp bumbled into it. The tentacles shot nematocysts into the shrimp, stunned the shrimp. And then the submersible pilots and observers sat there for 40 minutes watching the anemone tentacles slowly grab the shrimp and pull it inside the mouth, which is in the center of the cylinder, and completely engulf the thing. So 40 minutes for one of these guys to eat a shrimp. I don't know how long a shrimp diet will last. I bet if you eat a shrimp, you probably don't need to eat many shrimp in a year if you're an anemone, because they just kind of sit there.
What rate are you from here?
There's a little bit of bacterial mat. You see the flocculent just behind some of those anemones, and warm water just wafting through. These anemones, there's a good chance these are all clones, all related to one another. They bud off and form babies that then grow up beside them. So when you see dense anemones, they're probably all from the same genetic grouping. That's something that some of the folks in our lab work with Cliff Cunningham at Duke University. So on the main campus, Cliff and one of the students up there is studying that very question, whether these anemones are a single clone or multiple. Also on these rocks you see little tiny brown nubbins that are a new species of Provenance snail. I believe it's still undescribed. If you remember it, if you've been watching at the Von Dam site, there are white snails in this kind of habitat at 2,500 meters, and down here at 5,000 meters. We're seeing a different grazing gastropod. They're quite small, a couple millimeters.
33 and a half right now.
What?
So these BB shrimp are about 20 millimeters on average smaller in total length than the shrimp at the Von Dam vent fields. So anybody who has ideas about why they should be stunted, or if they're stunted in growth, or other things we can look at, we're puzzling about why this should be so. We've checked with using genetics to see if they're different populations of shrimp. They are not. They're the same population. There's plenty of gene flow between the two sites, Von Dam and Piccard, or BB Woods. So we know that there's shouldn't be, well, at least for the genes that we've looked at, that they're the same. So that's not the reason why they're small. So we've been thinking about whether there could be a metal toxicity or some other kind of chronic toxicity that might stunt the growth of these shrimp. They are the same. I plotted the size distribution of these shrimp and compared it to what we saw in January 2012 using our collections from just a couple of days ago here, and they are the same. So these have been small in 2012. They're small again in 2013, and we don't know why. Okay, well, we've been browsing around with the video. We've been sampling fluids, and my understanding is this is the last sample, and then if we can squeeze out a little bit of extra time, we're going to try to pick up some more shrimp, and I will do gut content analyses on these small shrimp and let you know if they have bacteria inside their guts or if they are carnivorous, like some of their relatives down at the Von Dam field. Okay, so Casey has set us up for some really fine anemone video. Sure, please. So these anemones are, we think they are probably Miractis, if they are indeed the same thing that we find on the Mid-Atlantic Ridge. Miractis was named after the Russian submersible Mir. So you've probably been getting a good dose of how we name things on the seafloor. Often it's whimsy, often it's in honor of the vehicles that have played such an important role in discoveries. Sometimes it's the pilots who help us collect things. We ought to have a Casey something or other here. It's part of the fun of being an explorer, being able to name things. So Casey's asking what the white part is, the really, really bright white bits on the anemone tentacles. I'm going to make up an answer because I don't really know. I don't know. I was going to say it might be reproductive tissue, might be concentrations of nematocysts, and maybe someone will tweet Julie or whoever is collecting responses to our YouTube video and give us the real answer, and we can tell you.
The other one was really pure, so exactly. We stopped it after 4 hours. It's funny,
So if you look right between the two big anemones, center screen, there's a little tiny, looks like an anemone. That might be what I was talking about. These are probably clonal animals, and that might be a budded anemone on the base of a stalk or just separating from the stalk. Okay. Do you want to take over the... Okay, we're finished with the water sample for Julie. And I think that was filtering, right? Filter. And we are going to we have only very few moments left before we leave the bottom. And are going to pick up the slurp gun and try to get some of these shrimp from both the dense and the sparse patches. And like I said, I'll be looking at their gut contents this evening.
I gotta get the GC ready. Conveniently enough.
No boat late tomorrow.
Van down.
Guess I can't talk tomorrow then.
No boat late on YouTube.
Okay.
So I think we have the stopper in the quiver this time. I don't know if word was passed through the video that we collected 65 shrimp yesterday, but only 5 made it back to the surface. There were some very clever shrimp who swam right back out the suction hose to freedom. So this time we're going to really trap them inside and expect a good haul. In some ways it's easier to have fewer animals because one can focus and really take the time to look and do proper dissections. So I at least benefited from the small number of shrimp that were collected last night.
Slurp on. Full power.
And this one 24, yeah?
And then zoom in. We'll see if we can.
What's really nice about this is that we are already well situated, stable. We don't have to hunt around. We can just grab these shrimp and get our work done and be on our way home.
Turn off starboard inner light. Alright, so just follow if you see any of the big bags of daddies over here.
Down in that lower right-hand corner, that kind of big whitish, or up just 2 above your, yeah.
So is it on?
Yeah.
Chris was saying he likes to come down straight on top of him. 1, 2.
One of the big ones here. There's one, any of these?
Right over to the right-hand side, and you can get any of those, that big guy and the other babies down there. 3, 4, 5, and then go into the swarm and take a couple from the dense.
And
The,
there's one more big one up there if you want that one.
nah, that's enough. And then right in the heart of the deep, really dense shrimp. Oh, yeah. That is good. That's plenty. Thank you. Beautifully done. Yes, sir. Yep. It's a wrap.
It's a wrap. There'll be sampling on the way up. Video.
All right. Nicely done.
So, the time now is 2:22, We're basically at the end of our time for our sampling. We are ready to start our 4-hour journey back to the sea surface. Seems strange to have the day already over at 2:30 in the afternoon, but it is Friday, so hell, it's the weekend. Except that we'll be back at 4 o'clock tomorrow morning, starting all over again. Because we just don't know how to have fun. Except that, well, this is pretty good fun. So the last thing we're going to do now, we're just stowing the equipment. And what we're going to do is ask Casey to set up the cameras so we can do a video transect as we rise up off the sea floor. So Casey, when everything's stowed, we'd like to assume the normal position with the cameras. We can do that video survey as we come up the first 100 meters, if that's okay with you. And I will do the usual thing for Brennan Phillips at the University of Rhode Island, who's a graduate student who's interested in studying what animals live above the different vent sites. So he's going to do a like-for-like comparison. We're rising at 25 meters a minute. And so from the depth we're at now, 4,960 meters above this area that's teeming. I'm just going to check. Leighton, you've been logging our coordinates all this time, haven't you? This is almost certainly a place that Max is going to love to come and drop a C2D over. When it's so teeming with life, it must have really good lipid signals. So as we work our way up through the water column, we know that if we can land a C2D in pretty much this location. Can we do a Doppler reset before we leave? Is there any harm in doing that?
We have no idea because this is the cluster we've made just sitting.
Oh, I see. We're bouncing around all over the place. Okay.
Yeah, well, we can still get
Our center of gravity for... I think
Yeah.
it's two separate... No, it's okay. It's two separate things. I think Leighton on the ship's DP can capture everything. And so he'll be able to do a central gravity, so you don't need to worry about that on yours. Yeah, if we can have center up a little bit more, so we've got the bio box just in the bottom of the frame, and I think we've been doing it with having the 2 inner lights on only, and not too much other stuff.
Well, do you want to make any video just coming up after
Oh,
this?
if we could actually, yeah, because that's really pretty. So,
I would just turn on all the lights except for the basket one.
so we'll have 2 separate things here. Leighton, there'll be a sample 25 is just going to be a scenic video, in which case let's get as little of the equipment in the view as possible. And then we'll switch to having the view with the bio box after that for zooplankton. Yes.
Can you write down because I'm looking at...
Yes, go ahead.
This is from waypoint 50 outwards.
Got it.
Actually,
Great.
Right. You ready?
Yes, thank you. So we're just going to do a little fly forward now to end this dive to go further into this valley. We've been sat down, as Casey was explaining earlier. It took quite an effort to get ourselves down and settled into this hole. But now as we drive forward, we actually get to see the full magnificence of this little rocky valley. You can see the boulders are pretty much the same boulders as you can see in the far field on the right, but just in this area where the fluid is flowing out. In the cracks you can see the rocks are actually a darker grey colour or pale grey, and then the shrimp living all over the place in here. Absolutely stunning. It's hard to see any focus because there's so much fluid flow. Shrimp living all around these crevices, and then the anemones that actually feed and prey on the shrimp all hinging around there. Absolutely stunning view. So yeah, if we could change our camera perspective now. So we just got the bio box just in the front of the view. And that's when we would like to switch to just the 2 inner lights on, please. And we're starting at a depth of 4958. And we're going to go up 100 meters. And I'm going to call out the depths every 2 meters until we get to 4858. And what Brennan's going to do is take the images from this high definition video at 30 frames a second and take the still frames so you can analyze to look for what zooplankton are actually living above a vent site. So coming up, we're now at a depth of 4956 meters. The light dropping away as we stop lighting the seafloor. It gets pretty dark pretty fast. We're now at 4954 meters. 4952 meters. 4950 meters. 4948 meters. 4946 meters. 4944 meters. 4942 meters. 4940 meters. 4938 meters. 4936 meters. 4934 meters. 4932 meters. 4930. 4928. 4926. 4924. 4922. 4920. 4918. 4916. 4914. 4912. 4910. 4908. 4906. 4904. 4902. 4900. 4898. 4896. 4894. 4892. 4890. 4888. 4886. 4884. 4882. 4878. 4876. 4874. 4872. 4870. 4868. 4866. 4864, 4862, 4860, and 4858. That's 100 meters above the seafloor. That's the end of our video transect. So thank you very much, Casey and Mario, for all your piloting and navigating today. Good job. That's the end of Nereus Dive 062. Short but sweet to the Piccard hydrothermal field. The time now is 2:33 here aboard the RV Falcor. We are approaching the end of our expedition, but we'll still be diving over the weekend. So please do plan to join us again tomorrow. We should be making for an earlier start, we hope, batteries permitting. I think we're ending with 50% of our battery power still in the tank, so we should have no trouble getting charged up and ready to go, as long as no nasty surprises await with the vehicle overnight. If we stick to our plan, we will be launching at 4 AM tomorrow. We'll be on the seafloor around 7:30, in which case we would actually get closer to 7 hours on the seafloor instead of the 5 hours we managed today. And we will be visiting new vent sites we haven't seen previously this expedition. So thank you for joining us, and thank you indeed, on top of everyone else, to Leighton Rowley, who's done a magnificent job logging all of our events today. He's the cruise coordinator here for the Schmidt Ocean Institute. And yeah, we'll be cutting our cameras soon to retrieve our optical fiber, and we'll be back at the seafloor tomorrow. Thank you.
This is Leighton here. I'd also like to thank Chris German for his wonderful narrative during the short dive today, and I'd also like to thank Casey and the rest of the Nereus team. Cheers, guys, for their piloting and support during this dive, and also Casey's wonderful commentary. Thank you, and we'll see you all tomorrow.